So welcome to this day of pain biology. In the background, you can hear the music of Sam Shepard and Floating Point, together with Pharaoh Sanders. Let me see if I can get the pointer working. Yeah, so I'm going to talk today about some of the work of the molecular nociception group and focus in particular how we try to exploit genetics to find analgesic pain targets and hopefully develop new drugs. And these are our heroes: Sidney Brenner, the greatest geneticist of the 20th century, and Pat Wall, who trained a whole cohort of preclinical pain researchers who still dominate the field to this day. And here is my most famous graduate student, Sam Shepard, who is on a flight from Brazil as we speak, presumably first class. And he made the top jazz album in 2021 with Pharaoh Sanders called Promises, which I was just playing. Rather great. So let's start off on a positive note. Can we use genetics to find analgesic targets and develop drugs? And the answer is yes, we can. And two of the people that have done this are sitting over there, Arman Akopian and Chi-Chen Chen. Arman joined me from Moscow, brilliant molecular biologist, and he made a cDNA library from sensory neurons from DRG, and he subtracted all the messenger RNAs that are expressed in a whole range of irrelevant tissues, with the logic that if we find genes only expressed in sensory neurons, some of them probably have something to do with pain. And you can see that Arman pulled out acid-sensing ion channels, including the target of manbalgins, which are strong analgesics, some structural proteins, an ATP-gated ion channel called P2X3, and a very exciting voltage-gated sodium channel, which we'll hear more about later in the talk. So Chi-Chen, who came from Taiwan, did his PhD focusing on P2X3. This is an ATP-gated cation channel, selectively expressed in sensory neurons. And 20 years later, a drug named after our head of department, Jeff Bernstock, the founder of the purinergic hypothesis, gefapixant, is actually now in the clinic in Japan, and GSK have just purchased a P2X3 homomeric antagonist for $2 billion. I didn't realize cough was that much of a problem, but hey. So this is an example. The average time between a preclinical discovery and drug development seems to be about 20 years. And we can discuss over lunch when libelous comments will be possible, why this takes so long. I should remind the speakers that this is being recorded. So libelous comments must only be made if they're extremely humorous. So this is what we know. There's a slide I've stolen from Dave Bennett and adapted a little bit. This is what we know about the molecular aspects of damage sensing in the mammalian system. It's pretty hopeless to focus on pain in the CNS because it's all basically circuitry and glutamatergic signaling. But there are lots of specific molecules involved in transduction of tissue damaging stimuli in the periphery. But there's an enormous amount of redundancy. So if you look at heat sensing, the group at Leuven who really pioneered the study of TRIPS have shown that there are 3 heat-sensitive TRIPS, so that if you knock one out, the others can compensate. And this facet of redundancy in primary transduction mechanisms is general. So Michael Caterina, who cloned TRPV1, showed that if you knock it out, you really don't get any behavioral effects on noxious heat sensation. This is also true of many of these GPCRs and receptors that are activated by mediators released from damaged tissue. Mechanosensation is an enormous problem, and there's still a big prize out there because we don't know how noxious mechanosensation works. Bert van Kost cloned these molecules piezos, and Niels Eichelkamp, sitting in the front row, showed that piezo2 plays an important role in mechanical allodynia. In terms of noxious mechanical pain, we still don't know how that works. So basically, transduction mechanisms are so redundant, it's not an exciting area really to focus on. But those mediators and immune products that change the gain of the system, like nerve growth factor important in osteoarthritis and bone pain, and TNF, which is a big deal in rheumatoid arthritis, these are interesting targets and are actually neutralizing TNF molecules in the clinic. So we really got very interested in voltage-gated sodium channels because these are the molecules that send the electrical signals into the central nervous system in response to these large range of different insults. And there are some sodium channels that are fairly selectively expressed in these neurons, and these are obviously interesting candidates to focus on. just to emphasize the complexity of primary transduction mechanisms, here are some experiments where we've compared animal models of different pain conditions, things like bone cancer pain, inflammatory pain caused by Freund's adjuvant, neuropathic pain with sciatic nerve ligation, and deep muscle pain by injecting acid into muscle. And you can see that the altered transcriptional profiles are completely different in these different models of pain in mice. This doesn't imply that these transcripts are significant in terms of the mechanism, but it kind of emphasizes the fact there's a multiplicity of cell types and mechanisms that underpin different sorts of pain. Now, if you work in pharma, you really want human validated targets. And so a number of people we're talking today are going to talk about heritable monogenic pain disorders. And again, if you look at these, it's quite intriguing that we get trypsin sodium channels popping up in these various syndromes. So TRPA1 causes familial episodic pain syndromes, excruciating thoracic pain. And then there are things like erythromalgia, which is a chronic inflammatory condition that's been linked to gain of functions in NaV1.7 and NaV1.8. And there's an interesting mechanical pain syndrome called paroxysmal extreme pain disorder that maps to NaV1.7. But really the most exciting aspect of this work is that there are some sodium channels that are linked to complete loss of pain, in particular NaV1.7. So you've all been to the dentist and you've all enjoyed the benefits of lidocaine. I love this story. So lidocaine, of course, blocks sodium channels, another reason to focus on them in terms of analgesia. So lidocaine was actually identified in a screen for blocking parasites that attack the beet crop in Sweden. I'm sure Patrick knows all about this. And accidentally it was found a small molecule that caused anesthesia. And so there was some medicinal chemistry carried out. And this was just around the period of the Second World War when poverty was extreme. And so 20 medical students were recruited and each one given a packet of cigarettes. This is magnificent. And it was shown that 1 mg per kg per hour of lidocaine application, was a very effective analgesic. The FDA approval followed with no statistical analysis because they said the effect was so obvious. Can you imagine this now? So here you have a very effective drug. This is developed probably for about $200 as opposed to $3 billion that we need now. So this also focuses attention on voltage-gated sodium channels. We know a lot about them now. We know there are 3 that are quite, or to some extent, selectively expressed in the peripheral nervous system. And then wonderful work from Nian Yang and Bill Catterall have shown using cryo-EM exactly how these sodium channels gate. So what I thought I'd do is tell you a little bit about the work of the people that have been focusing on NaV1.8 and NaV1.7 and introduce them because they'll be far too modest to tell you what they did themselves. So as I mentioned, Arman made this brilliant difference library. He discovered NaV1.8 and then he knocked it out and showed that inflammatory and mechanical pain were completely lost in the mice that were otherwise normal. And this is amazing because obviously this implies no side effects if you can manage to attack NaV1.8. All of our work was done in collaboration with a brilliant electrophysiologist called Lucia Civilotti, who I no doubt will be classified as our Rosalind Franklin in some book in 10 years' time, the person who did everything and was exploited by vicious old white men. What do you think? Just wait for that to come out. I'm sure it will. Anyway, Lucia is a brilliant electrophysiologist and has sadly stopped working now. But she basically is the link between function and all the genetics that we did. So it was clear that NaV1.8 was very interesting. And Mohammed Nasser and I designed a Cre recombinase driven by NaV1.8, a knock-in mouse. And Mohammed decided that he would flox NaV1.7 and see what happened. I wasn't at all keen on this, so I think one can make the strong case that Mohammed Nasser is the father of the NaV1.7 pain field. It's just fantastic. Now, poor Mohammed has the misfortune to be a Palestinian. And in these racist times, his poor sisters are in Gaza, where his parents died. And he's obviously emotionally very perturbed at the moment with the way things are going. So he apologizes, but he couldn't come to the meeting. So we wish Mohammed well. And clearly, he's made a marvelous contribution to the pain field. So Mohammed demonstrated that if you take 1.7 out in the 1.8 neurons, which we know are important for inflammatory pain, you can block inflammatory pain and mechanosensation very effectively. And shortly after he did this, a group in China led by Professor Yang showed that people with erythromelalgia had mutations in NaV1.7. I think that, that's really very interesting. Then Dibhaj at Yale showed that some of these mutations resulted in gain of function. So this is a background to the human studies of Jeff Woods and James Cox. Trouble with the trains today, so I hope Jeff is actually going to show up. But Jeff was busy mapping microcephaly genes, and he came across a pain-free person in Pakistan and mapped the gene. This is in the early days of DNA sequencing. And so James and Jeff showed that people without functional 1.7 were actually pain-free. So this is obviously fantastic genetic validation, and we were very fascinated to see how this worked. So Mike Minette, a graduate student, found that opioid peptides were upregulated in the 1.7 knockout, and that naloxone could reverse some of the analgesia. This is an in utero deletion in mice or humans. And then Vanessa Pereira and Stefano Lignier in 2018 showed that actually you can upregulate the enkephalins without any analgesic effect at all. This is published in 2018, a paper that's been completely ignored by pharma for some strange reason. Yet naloxone, which blocks opioid-mediated analgesia, was still effective on the 1.7 knockout. So this mystery of support was sorted out by Tim Hutcho, who showed that the entire opioid signaling system is actually massively potentiated in the 1.7 knockout. It's not a question just of opioid peptides. And finally, Donald McDonald, who's talking later on today, showed that neurotransmitter release, glutamate release, was severely compromised in the NaV1.7 knockout. So just to give you a little bit of data, this is the information from Arman's paper, where the slowly adapting TTX-resistant current in sensory neurons is completely lost in the knockout, and we get this mechanical insensitivity and a loss of inflammatory pain. Now, there's a big problem for pharma in that there's some deficit in the heart and the conduction system that we found with Jaspal Kunar at the Hammersmith Hospital. So this is the work of Jing Zhao. This is strange because we couldn't find any evidence for NaV1.8 in the heart. There's loads of it in the dorsal ganglia, and yet it seemed to be a heart attack risk factor. So this has been, this whole mystery has been solved by Christofferson's lab in Amsterdam. So what he's shown is there's a cryptic promoter that drives the expression of the terminal 8 transmembrane domains of NaV1.8 in the heart. And so if you look at this chunk of NaV1.8, it has no activity at all. And if you look at the heart channel, NaV1.5, and co-express it with this inactive lump, then you get a massive increase in NaV1.5 activity. So this explains why NaV1.8 is linked to various cardiac dysfunctions, including Brugada syndrome, which is a syndrome where you drop dead, generally as a young adult in a mysterious way. So I think NaV1.8 is such a fabulous drug target. I hope you'll let me just bang on this slide about the history of it. So Arman cloned and expressed it and showed that the knockout had a specific role in pain pathways in 1999. John Levine, always ahead of his time, showed that antisense oligos could block the expression of the channel and cause analgesia. And we actually founded a startup called Ionix Pharmaceuticals to make NaV1.8 antagonists. Sadly ran out of money in 2004, went bankrupt, but we clearly were on the right track. We found some conotoxins with our friends in Brisbane that were very good analgesics and very selective. And various other companies were hard at it, like Abbott. But these cardiac issues frightened away people from NaV1.8. And of course, the NaV1.7 story was so seductive that most pharma companies stopped working in that area. The link with Brugada syndrome as well kind of finished off the project. And even though human erythromelalgia was linked to NaV1.8, so that's a human pain condition, this is a very unattractive model. I should say that because of this cardiac role for the short form of NaV1.8, that's why you don't find any pain-free people with a loss of this particular voltage-gated sodium channel in the same way as you do with 1.7. So because it's essential for normal heart function. So presumably Vertex weren't aware of these these observations when they developed their NaV1.8 hugely selective blocker, 548, which is now in phase 3. And we really do hope that it succeeds and that other pharma companies pick up on NaV1.8 as a really good analgesic target. Now, let's move to 1.7. As I mentioned, when Mohammed knocked it out in the 1.8 positive neurons, he could abolish all the inflammatory hyperalgesia associated with prostaglandins, CFA, NGF, and so on, and went on to knock out the gene in all sensory neurons and get a pretty much pain-free phenotype. So we've got in humans, we've got these erythromelalgia mutations, and there's a beautiful paper from Catterall, just come out in General Physiology, explaining how these mutations make NaV1.7 hyperactive. We've got this other condition, which is linked to inactivation. And then we have Jeff and James's story about no channel, no pain in people. So how does this work? Well, we initially thought that the loss of 1.7, which is the first voltage-gated sodium channel to fire off when cells are depolarized, which this is required to recruit 1.8, and the loss of this channel means that there's a loss of electrical excitability. And this is absolutely true in the olfactory system. So mice and humans without 1.7, they can't smell. And actually, the olfactory neurons are massively compromised in terms of electrical excitability. But clearly, in utero, other things are going on. I should say that 1.7 is all over the place in the central nervous system and the periphery. It's in the pancreas, and there's an important role for it in sympathetic neurons in heat sensing. So if you knock out 1.7 in 1.8 positive neurons or all sensory neurons, you don't get any effect on hot plate behavior. But if you knock it out in the sympathetic neurons as well, then you make the mice pain-free. So 1.7 is important not only in sensory, but also in sympathetic neurons in terms of the pain-free phenotype of the human mutants. This is my favorite slide from Donald McDonald, sitting in the corner. Donald showed that in the embryonic knockout, the nociceptors are completely normal. And this is a wonderful slide showing using GCAMP that you can get sensory neurons to fire off in response to heat. And if you had an inflammatory mediator like prostaglandin E2, you awaken silent nociceptors and also fire up the nociceptors that are still sensitive. So when you do this, add prostaglandin E2 to a wild type mouse, obviously gets super sensitized to heat stimuli. But if you do it to the knockout animals, the knockout animals are still completely pain-free, even though they show exactly the same behavior as the normals. They have increased responses with heat-sensitive neurons, and silent neurons are activated. So this is a fantastic mystery. And as I mentioned, Mike Minute had shown that opioid peptides were upregulated in male and female mice, no effect in 1.8 knockouts, but big effects in 1.7 knockouts. And naloxone could actually reverse a lot of the analgesia. But there's clearly a problem because here we're looking at the induction of enkephalin in a 1.7 knockout on a mouse that's lost a transcription factor, NFAT5, upstream of the PENK gene. And you can see we're getting the same levels of opioid peptides, but the 1.7 knockout is pain-free, whereas the NFAT5 knockout, which has the same amount of enkephalin, is completely normal. There's no analgesia. So we can't rely on the story of opioid peptides in terms of a mechanism for explaining 1.7 analgesia. But Tim Hutcho, in a paper that won the Surtoner Prize in 2016, showed that actually the opioid system is enormously potentiated. So here we're looking at the effects of fentanyl on protein kinase A activity in NaV1.7 knockouts in red and wild-type animals in black. Here's a 1.8 knockout, exactly the same as the wild-type in terms of fentanyl action. And you can see fentanyl is stupendously active in the 1.7 knockout. So it's not just the opioid peptides, it's a whole signaling system. So Donald showed using a glutamate sniffer that actually you need an enormous amount of depolarization to get glutamate to be released from the 1.7 knockout. And he also showed that this facet of behavior was reversed with naloxone, and the glutamate release would become essentially the same as a wild-type animal in the presence of naloxone. So neurotransmitter release within the central nervous system is the locus of the analgesia. How does this work? We just wonder whether sodium could be the second messenger involved in this. So we already knew that if we increase the sodium in sensory neurons, we actually downregulate the expression of enkephalin precursor messenger RNA. If we fiddle around with the intracellular calcium, absolutely nothing happens. And if we block for disgated sodium channels with tetrodotoxin, we can get an increase in preproenkephalin message. So there's a relationship between expression of these peptides and intracellular sodium. And of course, we all know that GPCRs are actually blocked by sodium, class A GPCRs, including opioid receptors. So we did some experiments looking at opioid effects on NaV1.8 currents in sensory neurons, and we changed the sodium inside the cell. So as we go from 20 millimolar to a nominal 0 millimolar, you can see that fentanyl has much more dramatic effects on blocking NaV1.8 currents. So it's possible there's some kind of link between the sodium flux that's going through NaV1.7 and this opioid-mediated analgesia. So obviously this doesn't mean a thing unless it's true in people. And we found, again, in experiments done several years ago, that a human NaV1.7 null could feel pain when perfused with naloxone. This is greeted with great skepticism, so we did it again with some other people. It's clearly true that deleting NaV1.7 in utero causes an increase in the opioid signaling system in both mice and humans. I'm just going to spend 2 minutes on this because I think this is the crux of the issue. So NaV1.7 is all over the place. There's a wonderful paper by Scott Sternson and Tiago Brava showing that NaV1.7 can integrate small depolarizations to cause action potentials in the brain. It actually controls weight in the hypothalamus and so on. It also does an important job in the sympathetic nervous system, and it's involved in things like insulin secretion. So it's by no means sensory neuron specific, like NaV1.8. So the embryonic deletion clearly activates the opioid system. And I would say to anybody who's watching our recording, if you don't believe this, we're very happy to give you the mice and you can repeat the experiments. But the opioid system is not activated in olfactory sensory neurons. There's a loss of excitability. But because the sympathetic nervous system works and these NaV1.7 null people are completely normal, apparently, have children, they're smart, there must be compensatory changes in the embryonic nulls. Now, if you do an adult deletion of 1.7 in mice, you don't see this opioid effect at all. You see a loss of excitability, just as you do in the olfactory sensory neurons. So this is a different mechanism altogether. And there's absolutely no conflict between these two sets of observations. But what I think this tells us is that drugs that act centrally on the release of neurotransmitters will have lots of side effects, and drugs that act on the sympathetic nervous system are also going to be a pain. So this is pretty tricky. But if we forget about channels for a minute and just think about this opioid-mediated analgesia that the in utero deletion has shown us, if we could just fire that up in some pharmacological way, that's fantastic. These are people that aren't addicted, they don't have constipation, they don't have respiratory depression, but they have a lifetime of analgesia mediated by endogenous opioids. So I think this is a great insight and an area where we should be working on. But I think channel blockers are not going to work. So I've got 4 minutes to go, and let me move on to talk about the work of Ryan Haroon, who's just arrived. I've lied to you. I've banged the drum for NaV 1.7 and NaV 1.8 as a sort of secret of treating pain, and it's just not true. If you look at bone cancer pain and you knock out NaV 1.8, you look at limb use and weight bearing, you can see these poor animals with cancer in the bones. The 1.8 knockouts are just the same as the wild types. And even the 1.7 knockouts, actually, there's almost no effect of knocking out these channels on this very vicious form of pain. But if you kill the NaV 1.8 positive neurons using diphtheria toxin, then you can see some useful analgesic effects. We published a story saying this wasn't true some years ago because we used far too many cancer cells. But if we use lower levels of cancer cells, there's a dramatic effect of cell silencing. So we decided to use chemogenetics, chloride channels that are activated by a modified nicotinic receptor in a hybrid form of channel developed by Scott Sternson. And so varenicline, an exogenous agonist, can activate these channels and switch off neurons. So we made a mouse with a flexed SAM4 glycine receptor. So if we have a CRE, we can express this in sets of sensory neurons and switch them on and off reversibly. And here you can see what happens if we switch off the NaV 1.8 neurons with varenicline. We can substantially block bone cancer pain. So this is really, I think, very exciting. If we look at neuropathic pain, chronic constriction injury, we have even more dramatic effects. We can basically completely ablate the pain. So this cell-based silencing approach is very attractive. But the problem is it's enormously expensive. And I think gene therapy is really not a runner until we manage to get to a situation where the costs are like similar to the Pfizer vaccine. You know, we're talking $2 million to treat pain. This is frankly ludicrous. But if you're talking $20, I think that's good. So in summary, NAV 1.8 is a fabulous drug target, and every pharma company should be working on it. 1.7, I think, is very difficult. Chemogenetics has a future when the cost issues are resolved. And just almost finishing on time. Let me say thank you to everybody in the group who did all this work, and thank you for your interest. And I just wanted to finish with a word from our sponsor. You know, Isaac Newton was one of the founders of the Royal Society, and I was amazed to read at the end of his Principles of Mathematics that he talks about electricity. He says all sensation is excited, and the members of animal bodies move at the command of the will by the vibration of this electric spirit, mutually propagated along the solid filaments of the nerves from the outward organs of sense to the brain and from the brain to the muscles. So Newton was clearly the first ever biophysicist and had insights into voltage-gated sodium channels in a kind of more general way. So thank you so much for listening, and I'm afraid I've had my whole half an hour. Thanks a lot.